All right, hello. Here I am with our last talk having to do with the nervous system, and I want to talk about sensory organs. Remember that we said that one of the important functions of the nervous system is to sense the external environment and send those signals to the central nervous system where they can be processed, and then the organism can decide what to do. So let's talk about how animals can sense their external environment. So obviously we're going to talk about sensory organs. So these are in the peripheral nervous system, right? They're going to collect data and send signals to the central nervous system. Now they're going to pass those signals to specific regions of the brain, and then that part of the brain is going to understand the signal. Right? So again, this is all just neurons passing signals among themselves. But somehow those things come together in a complicated way to create what we know as consciousness and to process these signals. You know, I'm not a neurophysiologist, so I can't speak to this in great detail, but the process, the propagation of the signal down a nerve and then the passing of that signal to other nerves, that's all pretty straightforward, and we've already talked about that. And so it's just how those things combine together to understand signals. That's where it gets complicated and very, very interesting. And so, of course, you can look and you can map these to your brain, and you can see how different parts of the brain process different signals from different sensory organs and put them all together to help to understand the signals that are coming in. And so there's lots of different types of sense organs. These are the broad categories I'm going to talk about. This is probably not an exhaustive list. But we're going to talk about chemoreception, mechanoreception, and photoreception. So chemoreception is just receiving signals and sensing chemicals in the environment. Excuse me. Mechanoreception is sensing mechanical movement in the environment, like the movement of water or the movement of air. And photoreception is sensing photons of light energy. So let's talk about these in a little bit more detail. So chemoreception is sensing chemicals in the environment. This is basically taste and smell, okay? And you know that the sense of taste and smell are sort of linked in us, right? You know, you can smell something, but you can also kind of taste it if it's noxious, or if you're eating something and it doesn't smell good. You know that those are linked. Well, they're linked because they're basically the same thing. It's a molecule in the environment that binds to a receptor, and then that receptor changes shape and depolarizes a neuron and starts a signal, and that signal, the shape change causes ion channels to open, which causes a nerve to fire, and those nerves go to a certain part in your brain, and that signal gets processed and you understand that signal. And so it all starts with a chemical or a molecule binding to a receptor and causing that receptor to change shape. So you've got to have a receptor that's the right shape for that molecule. And so, that's why you can smell some things and you can't smell others. You can taste some things and you can't taste others. Because if you don't have the proper shaped receptor for that molecule, then that molecule's got nothing to bind to, and so no nerve is going to fire. But again, think at its basic level, what causes you to sense that if you smell something? When that protein binds to the molecule and changes shape, that opens a sodium channel, or any ion channel. Usually it's an excitatory, so it's a sodium channel. Once sodium rushes in, changes the voltage, causes other sodium channels to open, and you have an action potential. Cool. So there's a figure from your book, kind of zooming in on, say, taste buds on your tongue. And so if you zoom in, you can see the different levels that they're looking at here. But basically, when you get down to the molecular level, you've got receptor proteins on the surface of these microvilli, so small, very small finger-like projections, lots of surface area. And when that molecule binds to that receptor, it changes shape. And you see that at the other end of this cell, you've got a synapse. And so by changing shape, you can depolarize a membrane, which can pass a signal on through the synapse and cause a nerve to fire. And another figure from your book showing a similar thing in the nasal cavity. And so again, you've got those microvilli, those very small finger-like projections that are covered in these different receptors. And if the right receptor is there for the molecule that's in the environment, that molecule can match up to that receptor, change shape. And again, if you look at the other end, up opposite from the microvilli, you can see an axon, you can see nerve coming in. And so if you open up enough channels to cause sodium to rush into that nerve, you can cause a nerve to fire. And then that nerve passes its signal to other nerves, and it goes to somewhere in your brain, and you can process that signal. And oh, that smells great, or oh, that smells awful. And of course, you know, it's many molecules binding to different receptors, causing different neurons to fire, that all sort of come together and is interpreted somewhere in your brain. And so again, as the receptor changes shape, it can cause channels to open and ions to rush in. And usually you're trying to, you know, I don't know that there's any kind of real inhibition here. You're just mostly you're looking at excitatory neurons that are allowing sodium to rush in. And so just one example of this can be found in pheromones. And so this is just kind of a side note here. You probably heard of pheromones before. It sounds a lot like hormone, but a pheromone is like a hormone that is released out of the body. It's a chemical signal, but they're usually species specific, and these are molecules that you can pass a signal from one individual to another. But you have to be able to sense that pheromone in the environment, and that's where chemoreception comes in. And so if you've ever, you know, seen a line of ants walking across the ground, this is how they're staying in line, right? There's a pheromone trail that they're able to sense, and they just follow that pheromone trail. And of course, there's lots of uses for pheromones and lots of ways that organisms use pheromones to signal others in their own species. Now, here's a video I have, and this is an example of an alarm pheromone. And so this is a student I had did this for her thesis research. She was looking at alarm pheromones in a species of fish called red shiner, and it's a common native fish. And basically these alarm pheromones are ways to signal other individuals that there's trouble in the environment, okay? And so what we have here is an aquarium you're looking at, and they're kind of hard to see right now, but once the video starts, you'll see in the lower right corner of this video, there's a school of about 5 of these red shiners, and they're all kind of schooled up, and they're all just kind of chilling, okay? And if you look in the back left, you see a pump coming in, and what she has here is she's got this tank isolated. It's surrounded by a box, so the fish can't see anybody. They can't see anything, and the camera is just sticking through a hole in the box. So the fish can't see anything. So they're not startled by people walking around, and we're walking around quietly, so you can't hear anything. And what she's going to do is she extracted some of this alarm pheromone from other fish, and she's going to inject it into that pump. And so then that small amount of chemical is going to get spread throughout this tank. And so see if you can tell when she injects that pheromone. See if you can notice a change in the fish behavior. So I'm going to play the video now. And you can see the fish in the lower right corner. Did you notice any change there? Look at what happens when she injects that alarm pheromone. They immediately sense it, and they immediately start freaking out.
And so this is what she found, and this is known in several fish species, but it's never been studied in this fish species. But what she showed is that you get an initial rapid sort of freak out. You get this rapid movement of fish in response to this pheromone, but then within 30 seconds they calm back down, and they don't school as tightly as they did. So she was measuring how close they were to each other before the pheromone, and then they have this freak out when she introduced the pheromone, and then after, when they calm down, they don't school as tightly. And so it's some sort of behavioral response, again, because they're responding to this chemical signal in their environment, and then that causes them to behave differently. And presumably this alarm pheromone is a way to signal, hey, there's trouble, there's a predator, we need to, freak out and run away and then do something different because there's something wrong in the environment. Ultimately, all of this is caused because there's a chemical in the environment that binds to a receptor, that changes shape, that causes nerves to fire, that causes a change in behavior. That's what you're seeing at the molecular level here. Okay, that was chemoreception. Let's talk about mechanoreception. And so this is mechanically or physically moving the receptor causes a nerve to fire, triggers an action potential. So before it was a molecule, it was a chemical bond that caused the receptor to open ion channels and cause a nerve to fire. Now we are just physically pushing on it. There's no chemistry here, it's more of a physical thing. You bend or distort the membrane, and that is enough to open up channels. By just pushing on it, you can open up pores, or you can open up room for sodium or other ions to rush in that will set off an action potential. So where are you going to find stuff like this? Well, classic example is the lateral line of fish, right? Another fish example, guilty. But you've seen the lateral line. Most fish have a lateral line. And if you look in this lateral line, it's a little canal, and you've got these tiny sensory hair cells that stick out in this lateral line. And those hair cells are kind of stiff, but when water vibrates, it pushes on those hair cells. And because they're stiff, that distorts the membrane of the cell. And when it distorts that membrane, it's like, stretching a mesh or something. It opens up pores. It opens up holes, which allows ions to run in. And if enough of those ions run in, it can trigger an action potential, because you can see it's connected to a nerve. And so that can cause the nerve to fire. And so something like, if a fish is nearby and its tail is beating, the movement of that water can move these hair cells, and those nerves can fire. And which ones fire? If a prey or another fish is right over here, you can't see it because you're a fish, your eyes are up here, but that moving water causes sensory hair cells on that side to fire. Something is right there. And so if you've ever seen a school of fish and they seem to be very well coordinated, this is one way they're doing it, because they can sense all the fish around them and how they're moving based upon the movement of these hair cells causing certain nerves to fire. Pretty cool. Another place you're going to find this in a fish is in their inner ear. So this is what you're looking at. This is a figure from your book showing the inner ear of a fish. And basically what you've got here is you've got this otic chamber. There's two of these. You're only looking at one. And you've got this chamber, and it's filled with a very dense liquid called endolymph. So a very thick liquid inside this chamber, okay? You also have very heavy bones called otoliths that just rest. They sit in this chamber, all right? Inside the inner surface of this otic chamber are more of these sensory hair cells. These little stiff hairs that when pushed cause nerves to fire, all right? And so basically, this is how fish can hear. Because, sound is just the movement of water molecules, right? And so when those water molecules vibrate back and forth, the fish is mostly water. The fish vibrates at the same frequency. But those otoliths and that endolymph, those are a little bit denser than the fish. They're a little bit denser than water. And so they have inertia. And so when the fish vibrates, those things don't move. They tend to, an object at rest tends to stay at rest. And so the fish sort of vibrates around the endolymph and around the otolith. And so what happens is that otolith and that endolymph bangs up against different sides of the inside of that inner ear. Well, what's going to happen? What's on the inside of that inner ear? Those little hair cells that are connected to neurons. And so by banging on them, you cause some of those nerves to fire. But what's causing them to bang? This fish is vibrating because there's sound waves moving through the water. And so those sound waves, by banging on the fish, by moving the fish and causing that endolymph and the otoliths to push on certain hair cells, you're causing certain nerves to fire in the frequency of that movement. And those nerves go to the brain, and you process that as sound. And so that's how fish can hear, with the same sensory hair cells. In the same way, it's also used for balance, right? Because if the fish is upright, where is that otolith going to be sitting? That otolith is going to be pulled down to the bottom of the otic chamber due to gravity, right? If the fish is upright. And so that otolith is always sitting on sensory hair cells at the bottom of the otic chamber. So those nerves are firing all the time if the fish is upright. The sensory hair cells at the top of the otic chamber are not firing, because if the fish is upright, nothing is pushing on those sensory hair cells. But if you turn the fish upside down, now what happens? Now that otolith and the endolymph pushes on those hair cells at the top of the otic chamber, which it shouldn't be. And the ones at the bottom aren't firing. So now different neurons are firing, and that tells the fish, oh, I'm upside down. I need to right myself back up. And when you get back up, now the correct neurons are firing again, and that's how you can tell. So that's how you can use this for balance. The same sensory hair cells are triggering neurons based upon the orientation of the fish. Well, does any of this sound familiar? Does any of this happen to you? Do you have any structures that help you hear and also help you with your balance? Your ear does the same thing. Is it a coincidence? It is not a coincidence. Because why? Because we have a common ancestor with these fish. And that common ancestor evolved this inner ear and passed it on to the fish and passed it on to us. And so it works in the same way. And so if you look at this figure from your book, that's what they're trying to show you. So here is the human ear. And so now you're looking at the ear that you think here, that's the external ear. And then inside there, you've got your eardrum. That's the middle ear. And then if you go in deeper, you can look in this figure, you can see the inner ear. And if you look at that inner ear, it has a very similar structure to the fish inner ear that I just showed you. See those semicircular canals? The same semicircular canals. And that's evidence that we got this structure from our common ancestor, because it looks so similar and it functions in the same way. And so your inner ear has these same sensory hair cells that, when pushed, cause a nerve to fire, and that causes a signal to go to your brain, and you interpret that as sound. What pushes those sensory hair cells? Now it's not water, it's air molecules pushing, and air molecules pushing on that eardrum, which then pushes on the ossicles, those little bones on the middle ear, which then translates that signal to the inner ear, which causes those sensory hair cells to fire in that inner ear. But in the same way, those sensory hair cells are also used for balance. And so if you're upright, certain hair cells in your inner ear
are being pushed on at the bottom, and they're firing, and you feel you're upright. But if you flip upside down, now different hair cells are being pushed on, and that's interpreted as your brain as, oh, you're upside down now, you need to get right side up. Now, here's that inner ear from a fish that I showed you, and compare it to the inner ear of the human. See the similar structure? See the semicircular canals? You have otoliths. Your otoliths are much smaller than a fish's, but you see how you got very similar structure. Well, what's the best explanation for that? Common ancestry. Now, those bones in your middle ear, if you look in the fossil record, you can see how those bones evolved from jaw bones of fish. So early fish had several bones that made up their jaw, and as they evolved into just one or two bigger, heavier jaw bones. And so those extra bones were no longer needed for the jaw, and those evolved into other bones that improved hearing. And so that part of your ear you can also see in fish, but in early fish it was jaw bones, but then in tetrapods and other invertebrates, those jaw bones evolved into these ossicles, these other bones that help you with your hearing. Now, if you kind of study this again this figure is zooming in on that human inner ear to show how these sensory hair cells can take sound energy, and by moving those cells you cause nerves to fire and you can hear something. And so if you zoom all the way in, you see at the very basic level of that inner ear, those hair cells vibrate, causes the membrane to open up, allows sodium to rush in, which causes a nerve to fire, and that goes to a part of your brain that interprets it as sound. So it's the same sensory hair cells. And so what pushes those cells when sound vibrates? It's that thick endolymph. That thick endolymph resists motion. And so, the thick endolymph takes the sound energy and transmits the movement of that air molecules into endolymph motion, which can push on the sensory hair cells. But also, again, when we talk about balance, the endolymph becomes important. And so you've got that thick endolymph, and because it's thicker and more dense, it has more inertia. It resists motion. And so as you turn your head, that endolymph doesn't turn as fast. And so you got those hair cells with this endolymph, and as you turn your head, you're moving those hair cells through that thick endolymph, which doesn't move as fast. That causes those hair cells to distort. When you push on those hair cells, when you distort those hair cells, that opens up the channels, it lets sodium runs in, causes the nerve to fire. And so that's, again, how that inner ear helps you with movement and balance. Because as you're moving, the endolymph isn't moving as fast, but that causes those hair cells to fire, just like happens in a fish. And so this helps to explain other phenomena which you may be familiar with. If you drink a lot of alcohol, that alcohol crosses the membranes easy, gets into the blood easy, and gets into the endolymph easily. When it gets into that endolymph, it changes its density. It thins out the endolymph. Now that endolymph can move easier and flow better than it normally does. And so if you're sitting still, that endolymph can flow. But when that endolymph flows, what's it going to do? It's going to push on those hair cells. So those hair cells are going to fire, right? But you're not moving. Usually, those hair cells fire when you move your head. But now, the endolymph has been thinned out by alcohol, and that flowing endolymph pushes on those hair cells even though you're not moving. But you've got other sensory organs, like your eyes, that tell you you're not moving. So your ear is telling you you're moving, your eyes are telling you you're not moving, you're getting conflicting signals, you do not feel good. This is a hangover. This is the spins. This is, a lot of times when you do not feel good is because you're getting conflicting signals from different sensory organs, and your brain does not process them well, and you feel sick. So this is why when you get an inner ear infection, your ear neurons are firing, but and telling you that you're moving, but maybe your eyes are telling you you're not moving, and your rest of your body is telling you you're not moving. Your brain cannot process those signals, or is confused by those signals, and you feel malaise. You don't feel good. So this is, again, by understanding how these things work, then you can start to understand other processes. Here's a cool example from your book. It's evolved in these moths, right? And a big predator of moths are bats. And in fact, moths evolved first and are very nocturnal, which is why moths are kind of drab-colored sometimes. But that's also why bats are a big predator of moths, because bats are also nocturnal. And consequently, a group of moths broke off and became more diurnal, became more active during the day to get away from the bats. But during the day, when the light's out, now it pays to be very bright and colorful to attract mates, and so this is where you get butterflies from. So that's a side story. But anyway, if you look at some of these moths, what they're showing you here is you've got these air sacs that are in a certain way, they're built in a certain way, and they're fine-tuned with these neurons, that when the echolocation, the ultrasonic air vibrations from a bat hits this portion of the moth, they are tuned to that frequency, which causes them to vibrate in such a way to, again, open up those sodium channels and causes these nerves to fire. And these nerves often kind of cause the moth to fly randomly. These nerves are processed by the brain, and they just go straight to muscles. And so whenever that ultrasonic vibration causes these nerves to fire, the muscles move in such a way that the moth just kind of flies it flies erratically, which makes it harder for the bat to catch it. And so it's all just by having this structure evolved that was tuned to the frequency that the bats use to hunt the moths. Pretty cool. Okay, so the last type of sensory organs we need to talk about have to do with photoreception, or sensing light. So we're talking about seeing here, right? So to talk about sensing light, we need to talk about the properties of light, right? Go back to your physics a little bit. Remember that light has the properties of both a particle and a wave. And so light exists in discrete packets called a photon. So when light's coming at you from a light or from the sun or from whatever, and it's hitting your eye, it's actually, technically it's made up of little pieces. And there's these little photons. And each photon has a particular wavelength associated with it. And that is related to how much energy that photon carries, right? And so you can have a photon that hits your eye, and if that photon has a wavelength of 400 nanometers, then your eye would sense that as purple, right? But a different photon that has a different wavelength would hit your eye, and you would sense that as a different color. But anyway, light exists in these discrete packets of energy called photons. So when that photon, there's billions and trillions of photons hitting your eye at once, but when that photon hits your eye, there's light-sensitive pigments that have just the right shape to absorb just a particular wavelength. And so if that pigment has just the right shape for a particular photon, it will absorb that photon. So that photon disappears. That photon is a packet of energy that all goes in to that pigment. So the energy is transferred to that pigment. So photon disappears, but again, energy can neither be created nor destroyed, right? The first law of thermodynamics, that energy's got to go somewhere. Well, all that energy goes into that pigment. Well, that pigment's a protein, and it causes that protein to change shape.
just like we've seen in other examples. If you can add energy to a protein and cause it to change shape, this is another way to add energy to a protein and cause it to change shape. It's just a photon that is a perfect, this protein is the perfect shape for this particular wavelength, and so all the energy from that photon goes into that protein and it changes shape. But again, we're talking about proteins changing shape, opening a channel, allowing ions to rush in, initiating an action potential. So at its base, it's just the same thing as all these other examples. It's just a different way to kick off that action potential. All right. So here's a figure again of your eye, a side view of your eye showing all the different parts. And so those pigments are found in the retina, in the back of the eyeball. And this is where the photons come in through your pupil, the lens focuses them on the retina, and then these proteins capture those photons, change shape, start an action potential. Those action potentials all get passed from neuron to the optic nerve that goes into your brain and gets sensed by your brain as, you know, an object, as vision, right? And so whenever you're looking at me now, when you're looking at anything, that's because photons of light are leaving your computer screen, they're hitting that retina, they're causing these proteins to change shape, which is letting ions flow into a nerve, starting an action potential which travels to your brain, right? Okay. Those pigments are found in cells called rods and cones. Rods are for low light purposes where you don't really need color. Cones are for highlight purposes where you do need color. So the easy way to remember it, cones are for color, right? They both start with C. And so those rods and cones are what are found in that retinal layer. Let's zoom in on the retina. And so that's this figure from your book, showing you the relationship to the rods and the cones. You can see here. You can also see some nerve fibers and other things. Now you see the very back, you have chromatophores. Remember chromatophores? Chromatophores are cells that contain pigments. And so this is what gives your eye, you know, people that have different colored eyes, it's these chromatophores that have different colored pigments, right? But I don't think these pigments are, these are not what's initiating the electric signal, the nerve signal, right? These chromatophores are just giving color and they're helping to collect light and reflect light back into your eye, but it's the rods and the cones that are actually absorbing the photons and starting an action potential. Okay, so look at where the rods and cones are. And so they're showing you the rods and cones here. And then ganglions are nerve cells, right? And so these ganglia are the nerve cells that get stimulated by the rods and cones. And when they get stimulated enough, they send an action potential which carries that signal to the optic nerve, and your brain interprets this as vision. Now, I just want to take a side here and point out the really poor design of this, right? The nerves are at the surface of the retina, and the rods and cones are deeper in the retina. The rods and cones are the things that need to capture photons, but they're not at the surface where the photons hit. The photons hit the nerve first and then have to go deeper until they hit the actual rods and cones, right? This is a terrible design. If you were designing an eye, you would put the rods and cones on the surface where they would hit the photons first. This is just another example why we think, you know, that these things evolved from earlier versions, and natural selection just tinkers with those earlier versions and does the best with what it has. So again, we have this classic confrontation in biology, you know, where some people would say, oh, all things were designed by a supreme being. and the Supreme Being designed everything, and that's what we find on Earth. That could be true, right? But it's not a scientific explanation. A scientific explanation can't rely upon the supernatural, it has to rely on the natural, right? And so the natural explanation is that something like this eye evolved from earlier ancestors and tinkering with proto-eyes in those earlier ancestors. And if you take my evolution class, we talk about this in great detail, how we can see the evolution of the eye over time. But you can look at this and say, well, if you do believe that things were actually designed by a Supreme Being, this is not a good design. If you were a Supreme Being and you could design an eye, you wouldn't design it like this. And so this doesn't mean that there's no God. This doesn't mean that there isn't a Supreme Being. It just doesn't look like these things were designed by that Supreme Being, because if this is a really poor engineering, this is a really poor design, because the rods and cones are not located in the right spot. This doesn't mean you have to give up your religion. It doesn't mean there's no God. It just means that probably a better explanation for this design is that it's just the result of natural selection over many years. And that's all it means. So, take it or leave it. But that's kind of the scientific explanation, is that this evolved from earlier eyes. And that's why it's not, a perfect solution, but it's a good enough solution. Another bad design to our eye is look at where the nerve is. And so the signals from the eye gets carried to the optic nerve, which exits through a hole right in the back of the retina, right? So you've got a blind spot right there where that nerve is. You can't have retinal pigments there because the nerve's got to go through. Again, if you were designing an eye, you wouldn't design it like this. You'd figure out a better way to get that signal to the brain without creating a blind spot. So, the scientific interpretation is this was not designed by someone. This is just tinkering. And, it's a pretty good result, but it's not a perfect result. Anyway, let's talk more about those rods and cones. Like I said, the rods are used at night when you don't need color in low light. The cones are during light, when there's more light and you can use color. And so you use them at different times of day. Sometimes you're dark adapted and sometimes you're light adapted. Well, of course, you've experienced this. If you're in a dark room and someone comes and shines a flashlight in your eyes, you can't see for a second. You got to blink. If you're in a dark room and you walk out into the sunlight, you got to blink a little bit and you can't see very well until you've adjusted to the light. Or if you're outside and you walk inside and it's very dark, you've got to blink a little bit. You got to look around. You can't see very well because you're either using rods or cones, and then you're going quickly to an environment where you need the other one, and you've got to switch. You got to switch between dark adapted and light adapted. And so this is kind of showing you what is going on in your eye during that process. And so this is low light versus high light. And so on the left, you've got low light intensity, you've got nighttime or dark, and on the right, you've got high light intensity. Now, look at this, you'll see similar structures, you see the same cells and everything, but realize that this is flipped relative to our earlier diagram. So now, in our earlier diagram, the light was coming up from the bottom. Here, the light is coming down from the top. So just realize that, but you can see that everything is still relatively in the same location. So if you look at the low light intensity on the left here, you see the light's coming in, but not very much light is coming in. You see that the rods are pulled up closer to the surface of the retina. So that way the rods are more likely to get hit by photons than the cones. That's because you need the rods at low light. And so at low light, the cones are retracted into the pigmented layer. They're pushed farther from the surface of the retina, farther from the photons, because you're not using them. The rods are more exposed. But if you look on the right, at high light, you see it's reversed. You see the rods get retracted into the pigmented layer, and the cones get pulled closer to the surface of the retina, because you're going to need to use those cones in highlight. So that's what's happening when you're light adapted versus dark adapted. And so if you're in the dark, your rods are all close to the surface of your retina. And then if you walk out into a bright sunlight and you can't see, those rods have to be pulled back into the pigmented layer, and the cones need to be pulled up closer to the retinal layer. But that takes a few seconds and a few blinks until those have moved back and forth, and then your vision kind of gets restored. So anyway, that's kind of what's going on with your eye. There's a cool Mythbusters that I show sometimes where they test, their theory is, why do pirates wear an eye patch? And their theory was, well, because when you're a pirate, you're out on the deck in the bright light, but then you jump in and you're fighting and you go below decks to fight some more where it's dark. By wearing an eye patch, you have one eye that's light adapted and one eye that's dark adapted. And so when you jump down in below decks where it's dark, you switch the eye patch, and then you don't have that momentary lapse where you can't see. I don't know. They do actually kind of a cool experiment where they kind of suggest that might be true. So that's a Mythbusters, and if I can find the link, I'll post it, but I don't know. Just if you're watching Mythbusters, you'll see it sometime. It's pretty cool. Anyway, final thing I need to point out about this is that the cones are for color, and there's 3 types of cones in humans. Each of those cones absorbs a different wavelength of light. And so by having those 3 different types of cones that each absorb a different wavelength of light and combining them, you can get trichromatic vision, and that's what we have. But then you look at other animals like dogs and cats, they only have 2 types of cones, and so they have dichromatic vision. And other animals only have 1 type of cone, they have monochromatic vision. So when people say that dogs can't see colors, that technically they can a little bit, they just can't see as wide a range of color as we can, because we have 3 types of cones and they only have 2 types of cones. So there you go. Okay, so that is the end of our section on the nervous system. We got into a lot of details, but hopefully we talked about a lot of interesting things, and we explained a lot of things that you're familiar with, now that you understand at a deeper level. So that's all I have. As always, let me know if you've got any questions, and I'll talk to you later. Have a great day.